Hi everybody, so today we're going to talk about viruses and vaccines. This video is going to be split into two because there's a lot of information and I don't want you to have to sit there and watch the whole thing all the way through, so you can watch it in two segments. So we're going to talk first about the big questions. What are we trying to get out of this particular information? And so what we're trying to get is what a virus is, how the virus functions, what are vectors for the virus, and how do vaccines work. So we'll talk in this particular video about what a virus is and how they function. And then the next video will be the other parts, what are vectors and how do the vaccines work. So let's talk about what a virus is. The textbook definition of a virus is that it is an obligate intracellular parasite. What does that mean? Basically, to kind of quote Sir Peter Medawar, who's a virologist, it's a piece of bad news wrapped in protein. Parasites, as you know, they attach to a host and do bad things to the host, hurt the host, harm the host in some way, leaching information, food, or other things from the host. A virus is that. It's a parasite. The difference is it's a parasite with bad information inside that it's going to inject into the host, and the host is then going to have to deal with that information. It is hidden from view with what's called a protein coat. And a protein coat, or a capsid, is what you see here in this picture. It's like the purple or the green that you see or the blue that you see. It's a coat that hides it or cloaks the virus from the cells that it's trying to infect, so the cells doesn't realize that it's a bad thing. So here's different shapes, and there's different shapes for the things that it's specifically trying to target. We'll talk about that in a few minutes. The biggest thing is that viruses are non-living organisms. They attach to the host for the purpose of reproduction because they cannot reproduce on their own. They're not a cell. So they're not living because of that reason. Not that it makes a huge difference because they still are very dangerous, but they are considered a non-living thing. They've been discovered for all domains of life. Some are harmless, some are extremely deadly. It just kind of depends on what that particular virus does. The different types of viruses that we know about, we have Ebola, which we've heard of before. This right here is a photograph of those that are trying to protect themselves from influenza, which is one of our more common ones, or the flu. Here you have a child who's got little measles. Measles is a viral infection. This is a young man who has polio, another viral infection, and then of course smallpox, which is one that we've, I'm sure, heard a lot about over the years in history classes and whatnot. And so we have all of these different viruses. There's so many different viruses. I mean, it's equivalent, I think they said, to the number of stars in the universe. There's that many. But these are the ones that we really spend a lot of time dealing with. So let's talk about the types of viruses that exist. There are two types. The first of these is what's called a prokaryotic virus. And prokaryotic viruses, so that you understand, you have prokaryotic cells and eukaryotic cells. Many of you know that. Prokaryotic cells are those that are single-celled bacteria, single-celled bacteria. There we go, I got it out. Single-celled bacteria that have information, DNA information in the center of it and some proteins throughout it, but they're not organized in a real straight organized way like you would see in animal cells, or what we call eukaryotic cells. They are infected by viruses, and the viruses that infect them are what are called phages. That's the term that's used, and you see that written right here. They're called phages. And they're unique to bacteria because they're built in this way, and when we see schematics of viruses, this is usually the one that we see, and it's what's called a bacteriophage. And the purpose of this shape is that you have this protein coat up here, and you have these little protein legs that make the cell recognize it as what it would think is another cell. And so it goes through these two different types of life cycles because of this recognition process. So we're going to talk about these two life cycles. You have what's called the lytic cycle, which happens in 4 steps. You have infection, where it injects the information that it has into it. It makes the cell replicate that information, making new pieces of that bacteriophage, and then it assembles those pieces into full bacteriophages, and then the cell erupts, and those new created bacteriophages will be released into the cell, into the atmosphere, or into the body of water that it's living in, and then it will go on to infect the next ones. So this one you can see is kind of damaging because we have multitude of these viruses that just keep getting made. The difference is it's easy to stop this one because it is only infecting one cell to start with, and then it has to go to the next cell and the next cell, so it takes a little bit of time, and we can usually help the immune system in our bodies. If we were to go through something like this, we'd be able to attack those kinds of phages before they got too far. Unfortunately for bacteria, they have to sort of live with it until eventually they go away. The other way that bacteria or phages replicate is through a process called the lysogenic cycle. And the lysogenic cycle also has 4 steps. It has attachment, same thing, what's called penetration or again injection. But here's where it's different. It goes through a process called integration, where basically that genetic information gets integrated into the DNA of that cell, and then the cell stays alive. But the cell itself will undergo normal reproduction and replicate, and then every cell that it replicates makes new ones. And these are the ones that can infect us as humans and cause a good deal of problems, because what happens is this bacteria is now tainted bacteria. And so a lot of times for us, if we have antibiotics that kill bacteria, if you have one that's got this mistake in it, you're going to have a problem with being able to effectively eliminate it. So this is called the lysogenic cycle. So you get the idea of how these things replicate. So basically, there's a strand inside of this virus that either has DNA or it has RNA in it. And so when we talk about the next type of viruses, the eukaryotic viruses, which is all of your multicelled animals, multicelled organisms, including plants, these eukaryotic viruses have a problem with synthesizing this DNA or RNA within their body. DNA is double-stranded, of course, and RNA is single-stranded information. It's inside of this virus, and the virus has that same protein coat, but when it's eukaryotic viruses, they put this thing called a lipid envelope around it, which is a layer of fats or lipids that make it extremely recognizable by our cells, thinking that it's something normal or good, and that leads to a huge problem because our cell is like a cloak, like an invisibility cloak, and so our cells will envelop that information, and then we're infected. And so that's where a lot of problems with eukaryotic viruses come in. So if we look at what's called a DNA virus, for example, you have this virus, it's got DNA information, it comes into the cell, and it releases that DNA into the cell. And it integrates that DNA, just like you saw in the lysogenic cycle. It integrates that DNA into our cells, and then our cells make more of their cells, more of these viral cells, and then it releases those viruses out into the bloodstream, which then attaches to new cells and keeps going until we can deal with it. And usually what deals with it is our immune system. The RNA viruses are similar in that when we look at an RNA virus, what we see is we have the capsid, all this information for the virus. It comes in, the RNA information comes in, and it sits in the outside of the cell, and that's in the area where our own RNA that we make gets copied for traits. So we go through and we make templates out of RNA. Well, here we're injecting this RNA, and our body recognizes it as RNA, and it makes templates of whatever that strand is, which happens to be more virus cells. And so this one's a little bit more like the lytic cycle, in that it's making these templates and erupting out more viruses through that process. So both of them are kind of similar, but the difference is, of course, how they infect us. There are different classes of animal viruses, so I'm going to put this up, and you can make this bigger if you want to. You can search for it too a little bit more. This just gives you an idea of what viruses we are in contact with frequently that are considered RNA versus DNA viruses, and it shows you a little bit about how they infect things. So there's many different types of viruses out there, but these are kind of, again, more common ones to us. So let's talk a little bit about how viruses work in terms of looking at 2 different diseases and sort of figuring out what the process is and what some of the problems are. So we're going to look at the 2 big ones, influenza and HIV. And I picked these 2 because one of them has a vaccine and one of them does not. So we want to talk about why. Both of these diseases are called an RNA virus, which we just looked at. And RNA viruses, of course, it injects the RNA and then it makes copies of the templates of that RNA. The problem with something like influenza, with flu, in our bodies, we make RNA strands and we use those strands to make traits. What we do, though, is our bodies have a really good checks and balances system in place for any mistakes that are made. So we have less of a chance of mutation. Viruses don't have that. They don't have the checks and balances in place. So when they make these strands of RNA, there's a ton of mistakes in them. And so what happens is, because of those mistakes, it's able to mutate really quickly. And so for us, we can't make a permanent immunity. Vaccines have to change every single year to match the fact that these flu viruses are constantly changing because there's none of that mutation in place. So the RNA comes in, it does its thing, and we make new viruses out of it. It's called an aerosol virus because it's transmitted through the air. And again, it is vaccinatable, but we have to constantly keep up with those mutational changes. The HIV virus is different. HIV is a problem. It's a retrovirus. It's still an RNA virus, as I said, but it uses a process called reverse transcriptase. So what happens is that RNA gets into the body, but instead of making more RNA, it actually turns around and makes DNA. And then that DNA ends up getting spliced and integrated into the cell's DNA. And then that cell permanently has this disease. And this then replicates. These cells will reproduce and they will have the disease, and the more and more will keep reproducing. We can't really stop ourselves from reproducing. And so what happens is we can't vaccinate against it. We can't keep up with it. And we can't target those specific cells because we don't know which ones they are when it happens. So in other words, just kind of give you an idea, our RNA is making actual DNA instead of what normally happens, which is DNA making RNA. So this leads to a lot of bad news for us because it's very hard to defend against something like that. And this happens through body fluid transmission, which I'm sure you've heard about before. So these are the types of viruses, and these are sort of how viruses work. I'm going to save this video, and then you'll have a second video in just a minute that talks about vectors and what vectors are and how they work.